Okay, so lecture today is about immunisation, epidemiology, and population health. So we'll probably split into kind of two parts. First, immunisation, and then we'll sort of talk about epidemiology a little bit. Okay, so yeah, the first part is immunisation. So I mean, like, briefly, the whole purpose of immunisation is to sort of initiate a primary immune response so that when you're actually exposed to a pathogen, you can get your kind of really large active secondary immune response, which should hopefully protect you from getting the disease. So that's the whole idea in a nutshell. But I thought we'd just start with some questions. So if everyone could just sort of answer in the chat. So I've got some examples of vaccines down here, and could you tell me which one of these is a live vaccine? Okay, so that would be the MMR vaccine. I'm not sure people are responding, but I can't seem to see anything. So, and which of these is a killed vaccine? So yeah, people getting their answer is pertussis. I have to say that's not the one that's commonly used. I think in the UK it's sort of a acellular kind of subunit antigen type vaccine, but that is an example of a kill vaccine that can be used. So which of these is a subunit vaccine? That one's actually the hepatitis B vaccine. Which of these is a conjugate vaccine? So that's the Haemophilus influenza type B. So I did leave one extra answer, the tetanus one. Does anyone know what kind of vaccine the tetanus one is? It's alright if not. I mean, we're going to go through that today. So yeah, I'm seeing the right answer. It's a toxoid vaccine. So yeah, they just take tetanus toxin, like inactivate it, and yeah, inject a version of that. Absolutely. Right. So after that brief intro, I guess we'll go through different types of immunization. So just really briefly on passive immunization. So the whole idea of this is that you would give immunoglobulins or antibodies that are preformed, and you'd give them intravenously, and they can provide sort of immediate protection, which is really useful in certain situations. So if you've been exposed to certain viruses, and you're not vaccinated, you're not immune, then these can be really useful. But they are short term, because once they're clear from the body, you can't produce them again. Hence the term passive. So they're given but not produced. And another sort of example of passive immunity, I guess not so much immunization, is the transfer as antibodies through the placenta from the mother to fetus, and that's really important in protecting the fetus when its immune system still isn't quite developed. But the bulk of this talk is going to be about active immunization. So I've just listed the different types here with some examples, which you can sort of go through in your own time. But the ones we're going to focus on are the live vaccines, the killed vaccines, the conjugate vaccines, and the vectored vaccines. I'll briefly talk about the subunit inactivated toxin one now, but there's not too much to say. So the subunit one, they just kind of pick a single subunit or antigen, and then they'll pick an antigen which they know is fairly immunogenic and will generate a sort of protective immune response, and they'll just inject that. An inactivated toxin, so that classic example is tetanus, because the way that tetanus causes disease is by producing the toxin. If you can neutralize that toxin, if your immune system produces antibodies against that toxin, then it can't really cause much damage, and you kind of neutralize the pathogen. But like I said, we'll go through the other types in more detail. So yeah. The first one is the live organism. And you can kind of have two slightly different types of this. You can have the attenuated form. So you get the original organism, but you you sort of treat it in a way so it can no longer cause disease. It's still alive, but it doesn't cause disease, so it's attenuated. But the risk with that is that it can revert to virulence very rarely. A good example would be MMR. Not a reversion to virulence, sorry, just a good example of attenuated vaccine. And you can have these related organisms. So these are similar to the pathogen that's caused disease in human, but they're not exactly the same. So like BCG, which is related to TB, but it's not quite the same. So you can use that to immunize people. So the advantages is that these vaccines will contain multiple antigens, and so they'll generate like a stronger immune response with loads of different antibodies, and they really mimic natural infection quite well. But the disadvantage I've mentioned one already, you can revert back to virulence. Another big one is that you can't use them in immunocompromised because their immune system sort of can't control the disease at all. And they need to be kept refrigerated and kept cold. So you have to think about how you're going to transport them to different places. And kind of when they get to where they need to be, does that hospital or GP or whatever, do they have the resources to keep them at the required temperature? Because some of them need very, very low temperatures. The other type is killed. So you treat them, that can be chemically or with heat or with radiation, and you kill them. And so you sort of inject the antigens, but again, the pathogen can't cause disease. Big advantage is that these can be used immunocompromised, and they also don't need to be kept cold, and they're much cheaper than other vaccines to produce. But the disadvantage is that you often need boosters because you've killed the pathogen. You often have fewer antigens and so a weaker immune response. The next type is the conjugate vaccine. So I've kind of tried to illustrate here. Some pathogens, the major immunogenic antigens are polysaccharides and not proteins. And T cells can only recognize proteins. That means that any immune response that you generate in response to these pathogens is going to have to be T cell independent, which is possible, but you'd sort of rely on these kind of B cells. And they can generate like a fairly decent immune response in adults, but you wouldn't get much memory with that. And in infants, this pathway just isn't developed well at all. But if you sort of conjugate or add a protein, the T cell can now recognize this complex, and so you get a T cell dependent immune response, which is sort of like your classic immune response, with T cell help, and then B cells producing antibodies, and then memory B cells, and again, it's much better in infants. So these have been really useful in targeting pathogens like Haemophilus, which previously the vaccines just weren't very useful at all, especially in infants. And then we have the vectored vaccines. So for these, you'd sort of take your viral vector and you'd insert genetic material from a pathogen so that the vector can now produce an antigen from the pathogen. Then you'd use these to immunize somebody. And so the viral vector can sort of initiate kind of like a infection, really. But instead it's starting to produce sort of some pathogen proteins. And so as your body deals with the viral vector, it generates antibodies against, or say a T cell response, because this is more of a viral infection against the pathogen that you're trying to immunize against. And that's really useful. So the viral vector that you use, you pick one that didn't cause disease in humans. A common one would be adenovirus. And these can either just be live attenuated or they can be replication deficient. So that just means they don't replicate very well. But you do have to consider what viral vector you're going to pick. So if you picked adenovirus, there are certain strains of adenovirus circulating in the human population all the time. So you have really high levels of pre-existing immunity. And that means that the vector can be cleared really, really rapidly before you can generate an efficient immune response against the pathogenic antigens that you've introduced. So you might just pick a sort of a serotype of adenovirus or a specific subtype which doesn't circulate very much in the human population, or you might pick one which doesn't normally infect humans at all, but it still doesn't cause disease. That kind of makes sense. And the last really important thing to sort of think about with vaccines is adjuvants. And so you use adjuvants to kind of enhance the immune response. So they kind of typically stimulate innate immunity, and they sort of lead to some local inflammation, and that enhances strength of immune response. An example, those in clinical use are alum or aluminium salts. So that refers to like various types of these. And these are particularly important when you're using vaccines like subunit vaccines, which aren't very immunogenic. If you're using a live vaccine, they're less important, but they are a really important consideration when you're developing a vaccine. So that was all about vaccines. Any questions? Okay, I'm gonna move on. If there are any, just put them in the chat and I'll either address them as I go or I'll come back to them at the end. So another kind of few questions before we talk about epidemiology. I've got 3 definitions here, which I think are quite confusing, like between each other. So what is latent period for an infection? You can put A, B, or C to refer to the answers at the bottom. I haven't labelled them, but that's fine. Okay, I'm actually going to go through these on the next slide, but if you could put the infectious period for a disease as well, if anyone knows that. Again, A, B, or C. Okay, and final one would be the incubation period. I think that's where we were. Again, A, B, or C. So A, infection to symptoms, B, infectious to become infectious, and C, time during which the individual is infectious. Okay, so all the answers are on this slide. So first you have the incubation period, and so that's the time between when you're first exposed to a pathogen to when you actually develop symptoms. Then you have the latent period, and that's the time between which between when you get infected to when you are infectious. So that's kind of the time difference, if that kind of makes sense. And the infectious period is the time in which you're actually infectious. Is that clear for everyone? I think the difference between latent period and infectious period I find quite difficult, but like I said, so latent period between when you're infected to when you're actually infectious, when you can pass the disease on, and infectious period is a time during which you're infectious. And it's just really important to sort of think about how these are quite different. I mean, you can be infectious before symptoms, after symptoms, during symptoms. So I think it's really important to sort of get your head around these 3 definitions. Some more definitions I'm sure everybody knows off by heart. So outbreak. So this is a local occurrence of a disease in like a particular place at a particular time. And that's my little red dot on this animation. And then we have our epidemic, which is a larger scale outbreak, although it's still quite restricted. And then finally, pandemic, global. So enough said about that. Right. Sorry, there are a lot of definitions here, but I think it's really important to get them clear when you think about epidemiology. So then the attack rate is the number of new cases over the total number of population at risk. So this is our total population, and we get 4 cases. The attack rate would be 4 over the total, which I haven't counted. That's how it works. Then we have the secondary attack rate. And this is a measure of how infectious or the infectivity of a pathogen. And so it's the percentage of contacts who are infected who then develop the disease. So for example, we have our primary person being infected. So the attack rate will be 1 over the total. And that person then causes infection of 4 more people. So these are the people you consider when you're calculating the secondary attack rate. So I've written a few questions about these, which I think is probably just worth, you can do them after the lecture because it's just kind of maths. But one important thing to think about is back here. You have to think, when you calculate the secondary attack rate, you have to take into consideration that the orange person who was first infected is no longer one of the people at risk. So you have to kind of remove the initial infected people from the population. But I've written some questions that you can have a go at. Right. And then when you think about the R0 or the R0, so this is the number of people who develop disease after the contact with infected individuals. Again, so we've got our initial person in the orange and then 4 more people in the purple being infected. So if the R0 is less than 1, the disease will disappear. If it's greater than 1, the disease will continue to spread and the outbreak will get bigger. Again, I'm not going to label that point. I'm sure you've heard enough about the R0. Right. The next one to think about is the cycle of infection. So whenever you're sort of describing a pathogen, you can think about, or disease, I should say, you can think about the infectious agent, which is the actual pathogen causing disease, the reservoir, which is kind of a collection for the pathogen where the pathogen kind of likes to reside, the portal of exit, so how it leaves, mode of transmission, the portal of entry, and a susceptible host. And you can do this for pretty much any pathogen. So I picked influenza virus as an example. You can think about the pathogen being influenza virus, the reservoir being infected humans and possibly also surfaces, the portal of exit being a cough or a sneeze, mode of transmission, droplets, the portal of entry would be kind of the respiratory system, breathing it in, and the susceptible host is going to be a human. And whenever you're trying to break this cycle or chain of infection, you're just trying to stop it at one of these points. So again, with influenza, for example, you could make sure you wash your hands and clean any surfaces, and that will stop. It will kind of stop the portal of exit, kind of mode transmission, everything like that. You can cover your face when you're coughing and sneezing, again, blocking the portal of exit and transmission. And you could reduce the number of susceptible hosts by vaccinating everyone. Right, so a little bit of a change now. So just thinking about some techniques that you can use in epidemiology. So one of the ones that you probably heard about is the case control study. This is when you get 2 groups, one who has the disease, one without the disease, and then you're going to compare previous exposure. So this is a retrospective study, and it's an observational study because you're not actually changing anything. So the advantages are that this can be useful for rare diseases where you might not get enough numbers for a randomized controlled trial, and it can be useful as a starting point for epidemiological research. And it can also be useful if you're trying to investigate whether an exposure has a negative outcome, because it wouldn't be ethical to expose people knowing that there's a high possibility they get disease in the future. So you'd probably just use the case control study in that sense. And another really important disadvantage is because you're doing a retrospective study, there's a high risk of recall bias. So you might ask people to remember things they did 10, 15 years ago, and you might not get accurate data as a result. Next type is a cohort study. So cohort study, you start with the exposure, and then you look at who develops the disease. And this can be retrospective. So you're looking back after the disease has happened, but your starting point in your study is still the exposure. It's just the time in which you started the disease has already happened. And it can be prospective, which is when you follow people for a long time. It can be after the exposure. It could even be before if you're just tracking people. And then you're seeing who develops the disease. And the advantages and disadvantages are fairly similar to the case control studies. And then you have your randomized control trials. This is when you have your participants being randomized to 2 different groups. So if it's double blind, then neither the participant or the researcher will know the group, and that's kind of the aim for most kind of scientific and clinical studies. And yeah, so you probably want to use a placebo, again, to make it double blind. So when you've done your randomized controlled trial, you might be thinking of doing a clinical trial. So the randomized control trial can be used as part of a clinical trial, I should say. And so the whole pathway, if you're trying, for example, to bring a new treatment to clinical use. So first you'd have your preclinical studies, which are sort of carried out in vivo, so in animal models, and in vitro. Then you've got your phase 0 studies. So this is carried out on a small group of patients, about 10, and that's just testing the pharmacokinetics. So is it cleared from the body as you expect? Is it absorbed as you expect? And the pharmacodynamics as well. You then go into phase 1, which is a slightly bigger group of patients, probably 10 to 100. And this is a dose escalation study, so you gradually increase the dose, and you're really checking the safety at this stage. Like, is the treatment or drug that you're testing toxic? Then you move on to a phase 2, which is an even bigger group of patients, so this time hundreds. And in this case, you're really testing the safety and side effects again, but you're actually getting to looking at the efficacy in this case. Does it do what you expect it to do? Does it treat the disease you're expecting? Then you do a phase 3 trial, which is an even bigger group, so thousands, again, testing efficacy, safety, and side effects. At this stage, your drug might be approved, and that's great, but you're not done yet. You got to do your phase 4 trial, and this is kind of everybody who uses the treatment. If they have any adverse effects, they can report it. So it's just kind of continual monitoring to see if the treatment is safe, because I guess that's probably the most important thing, really. So that was kind of the end of lecture. I appreciate it was quite short. Any questions?